And so keeping the data life cycle in mind, I'm going to turn over the exercise to an exercise next to Deb Paul. And she's going to start from, okay, so we have a plan. We have started thinking about data lifecycle. How do we actually facilitate that? How do we actually map things to Darwin Core? And how can we actually start thinking about metadata in order to describe these data sets to make this plan something that is doable and actually easier on me as a researcher. Deb?
Hi, everybody. It's lovely to be here with all of you and see both familiar faces and names in the crowd. Hi. And to be meeting some of you for the first time. And I would like to start before I share my screen here real quick, just out of curiosity, and a +1 in the chat would be great for this. So how many of you already use Darwin Core or other standards from the Biodiversity Information Standards Organization, or EML, +1 in the chat, or other data standards for that matter? Very cool. Great to see all those +1s popping in the chat. Aha, working with NatureServe. Okay. Out of quick curiosity then too, where did you learn about data standards then briefly? So for those of you who had +1s to put in, where did you learn of their existence? Thanks, Link. Good to know. It's great to understand a little bit more about where we're coming from, and as Katja pointed out, when we tend to learn these things, how do they come to our awareness, and how that might change the practices, as Katja is talking about how we pass on this information. From my background, which starts with a master's in education before I came back to biology, we teach how we're taught. So we pass on what we learn, and unless we get exposed to something that causes us to take pause, maybe this is a thinking about how you gain new ideas, like when we share talk, like what we're doing here. So knowledge transfer is important. So thank you very much for sharing those. I might have a few more questions for you as we go by. Something to keep in mind when I give this talk, thinking about your data sets, how reusable by others are they? And you don't need to answer, and you're just answering your own head. And have you ever had issues with your own data where you couldn't make sense of it later? And so what are some of the things we're talking about here that could improve that situation for you and others and improve the impact that your research and your data have? So with that, I'll stop talking like this and share my screen here. And there we go. And am I on the right screen? Are you guys seeing?
We've got presenter view, Deb.
You do. Okay, let me switch it. Thank goodness there's only one button. Yay. All done. Thanks for your patience, everybody. 2 screens to set up with multiple screens to share is a little tricky. All right. So hi again. And I'm joining you from the University of Illinois, where I work with the Species File Group. Many of you know me for my 9 years with IDIG Bio in the ADBC network. I currently still work in the realm of biodiversity collections, more focused on the work of taxonomists and providing software tools that make it possible for them to do what they do and work the people they need to serve. So talking about the specific role that data standards play in the data lifecycle, think about your own data. And can you use data from last year or 10 years ago or 50 years ago? How many people can find that data? Where is it? So think about all the investment you spent as we talk about the upcoming slides. So we'll go briefly through what are data standards and specifically focus on Darwin Core with the point that standards really empower you and your research and create long-lived data. And we'll talk briefly about metadata because more people are going to talk about that after me. And we'll have a time for you to get in and try to map data. So we'll see how that goes. I see there's a lot of experience in the crowd, so that might actually go faster than anticipated, but we shall see. And then talk a little bit about what next. So many standards exist and new ones get developed when they are needed. Hopefully we don't recreate one. There's already one out there we can use. They are embedded in our everyday lives. So when we bring up standards, which ones do you think about when you think about standards? And how many of them are sort of, we just take them for granted? And as you can see here, they're, we are using them every single day. One of the ones we'll talk about specifically is the Biodiversity Information Standards Organization, known as TADWIG. I just finished 2 years of being chair of TADWIG. I had the honor of taking on that role. And I'm thrilled to be able to talk with you a bit more about that and answer your questions along with many TADWIG members in the audience. So there are standards indeed that are global. ISO 8601. And for those of you who know XKCD, you may have seen this. And many of you may have tried in your own data and recognized that through writing a letter or a number a little bit off, you couldn't tell if it was a 7 or a 1 or something like that. And all of a sudden you yourself were confused with your dates. Or if you're trying to use data from around the world and they don't store dates like Americans do. Sorry, Americans, I'm American. So it is indeed important if we want our data to be reusable and we want people to be able to do things like phenological research, we need to get the dates to be clear and unambiguous. So another reason we need standards, we all, and it makes sense, we keep data in the form that we need for our purposes. It's perfectly fine to have your lab standard and your collection protocol, have your data the way you need it to be. With your awareness standards, however, then you can make sure your data maps or aligns with the given standard so that other people who do the same, then your data can have an ability to be reused at new scales and new scopes and be understood by others now and in the future. And for many of you recognize this as the Rosetta Stone, where of course, including 2 things that the current people who could understand gave them the ability to go back and be able to read the hieroglyphs. So they provided the sort of standard, if you will, against which they could then figure out someone else's data. What is a standard? This is Wikipedia's definition. So notice the use of the word formal document and uniform criteria. So it's that, those help you, and along with the rules and definitions that are employed with each term, make sure that we put the semantic meaning in the box that we are supposed to put in there, and we also put the data in the format that's expected. And so now we can also potentially express relationships between terms in the standards as well. Metadata is the who, the what, the where, the when, and the how about the data itself. More about that in a minute. On the left, this is what we often see. We see data in different places, but not connected. So you have data in your own local data store, be that your local computer, be that your university data store, be that your computer at home, spreadsheets, a database that's not connected to other databases. So if you yourself wanted to pull data from those different sources, you would have to do a lot of work to get there. Once these data are mapped to a standard, and you only have to do the mapping once, then you have the data quality checks that you know have been employed about that syntax and semantics I was mentioning, as well as the ability to facilitate then data sharing and publishing and connecting across data sets who've employed the same standards as you. This then empowers more data analysis possibilities that weren't possible on the original scale or scope of the prior data sets, as well as enhancing the discoverability that Katja was talking about, or FAIR. These standards come from various organizations. We may not ever have heard of W3C. I encourage you all to Google it. It's very cool. You can thank them for things like HTTPS and www, the things that we expect to work when we use the web, as well as I just spoke about biodiversity information standards. This is a nonprofit group. It's international. Anybody can participate. You can come to us and say, I don't understand this standard, or hey, there's a bucket missing. I need a place to put this concept or this idea, and there's no term for that. And we'll be happy to have a task or work group get started on helping you with that. Put a +1 in the chat if you are a TADWIG member. That would be fun to see. I know there's TADWIG people here. Thanks. I'm looking. Oh, I see the numbers going. Ooh, very exciting. So briefly, TADWIG has interest groups and task groups. So interest groups talk around topics of interest, like people in biodiversity data, or identifiers for things, or material samples and how to standardize the data that comes from sampling events. You can join, and we do always use other existing standards when possible. So we try not to reinvent. We look first to see what standards are already out there. Learning more about TADWIG on Thursday, there's a 5-minute talk. You can click to learn more. Current examples, we've just talked briefly. Darwin Core comes from TADWIG, and it encompasses you sharing information about the who, what, where, when, and how. This could be about a given specimen that you are collecting, or it could be an observation record. If you want to share the metadata about how you did this entire collecting event, then that's a data set, and you can use the ecological metadata language standard. It's just one metadata standard, but coming out of the ecology community. And that's also who, what, where, when, and how. But at the level of what's inside this box, describe it to me so I know whether I want to look inside that box or not. The audio-visual media core is a standard coming from TADWIG about what's in your image, and helping you, the more metadata you give, then you can find the image inside that box that you need. And if you are doing sampling of a genetic genomic sort, standards exist to share that data as well that's related to a given sample or specimen. Diving down into Darwin Core quickly, we see it's divided into categories. So classes of terms, there's what's so-called record level terms. There are 19 of them. There are occurrence level terms, 19 of those, and I've highlighted them here. So we observe something in nature, or we collect something in nature. We would like to know who collected it, recorded by. Maybe we'd like to be a bit more specific, because maybe there are 2 J. Jones. So recorded by ID is something like an ORCID identifier for the who. So we can be very clear about who collected or identified something. Maybe we did sample for, we want DNA sequences. So this might be GenBank numbers here. And maybe we want to express relationships between whatever we collected and who was hanging out with it. So the bee on the flower, and whether it's host of, or eaten, or laying eggs on. This is where you can express those kinds of relationships as an example. And you'll be getting an exercise here in just a few minutes on trying to figure out if any of these terms fit with the data set for you to map. So then again, coming back, what about giant data sets that you have to go look at somewhere and decide if you want to use them? It's metadata that gives you and others the gift of sort of perusing that data to see if it's going to be worth your while to use. So it's really important for making data fair, findable, accessible, interoperable, and reusable. It's key to helping to make credit, citation, and attribution easier to accomplish, and even sometimes automated. So if this was the data set, which it is, that you're going to be looking at in just a second, what metadata would you want to have? There are some things here you can easily understand. You can see the where, you can see the state, it's Illinois. Is the date in the format that Darwin Core expects? I can tell you right now that the term year is the same in Darwin Core, but the term date is not. You'll have to go and look at Darwin Core and figure out, what do they call date? And do you notice any issues with the data here? So I'll ask you that in a few minutes. So EML, as I spoke earlier, is a metadata standard, and it comes out of work done by the Ecological Society of America. You can read a lot more about it, and there's going to be someone talking about that today, I think, in the very next session. Peeking inside a few, the EMLs are very rich and very detailed, and this is just the tip of the iceberg. On the left, focus on the box. So this would be you describing a data set. Who is the contact for the resource? Who created the resource? Who provided all of this metadata? And you can provide ORCIDs for those people as well. But you can tell us something about the how. What are the sampling methods, right? What was the project data design, the protocols that you used? What's the taxonomic and temporal coverage, as well as the geographic coverage of the data inside the box? And you can provide the keywords that you know your community would find appealing and want to search by providing the necessary keywords there. Just a quick taste of EML. Because this is what really happens, of course. You go out in the field with your project or your team or your lab, and you create some sort of piece of paper or field note or other document where you write down the what, the where, the when, the how, and any other observations that you have. You capture that somehow, generally, probably in a spreadsheet, and you name your columns some way or another. You might call lat for latitude. You might have an abundance column. You might have some sort of habitat description column. Then you've got to figure out where your data fits inside some other standard, in this case, Darwin Core. And the nice thing to remember about this is, while this does take work, it only has to be done generally once. And once you get that done, then you can continue to keep your data as you wish. And then when you share it or publish it, you put it out there, and you can put both if you so choose. You can put the raw data and the mapped data. So today, Brenna Decker and Alex Harmon-Treat volunteered to share their data set for the bees that they deposited as part of this work to look at how burns affect bee communities. I want to emphasize here that getting this bee data and the vouchered bees into the collections at INHS and Utah State University was a collaborative effort, planning in advance before the work took place, with the researcher talking with the collection manager, getting identifiers for the bees beforehand that were going to be vouchered, and working through and after field work to get these data, bees into the collection and the data out the door and published to GBIF. So this data set coming from here, what we're going to do is give you this data set to look at. And you'll be able to then look at each column header and see if you can find a matching Darwin Core term. Katja, is there anything you want to add at this point to?
No, I added a link to the chat. There's a lot of great conversation going on in the chat. I really appreciate what everyone's saying and sharing. I added a link to the Darwin Core mapping exercise, and the mapping exercise includes the well, you're about to go over it. I'll let you.
I am.
I'll let you do it.
But thank
Great.
you for jumping in, and thanks for everybody. I can see the number of chats, 34 in my box, but if I click on them now, I'll lose my place. So here we give you a link, which I think Katja has already given you, the Big B. Did you give them that link, Katja?
I did. I sent
Okay,
it
good.
in the chat, but I will do it
Cool.
again because the chat's going by so quickly.
Good. All right. So if you go to this link here, then you are going to find a document that looks like this on the left. And if you go to that number 1 link to narrative exercise, you'll actually see the kind of tutorial. It's essentially walking you through everything I just described. It walks you through opening this data set, looking at each column header, go to Darwin Core, see if you can find a matching term. So you can walk through that there. What you're going to do is open link number 2, unmapped B data. You'll see this. You have the option of, depending on your situation, you can either open that in your computer, or you can and use it in the cloud there, or you can download it if you so choose. And then what you're going to do, open this link to Darwin Core terms, because that's where you're going to find what you need in terms of, let me go really quickly here and grab that for you. We don't have that open in a separate window. Let me grab it. If you go to Darwin Core Quick Reference Guide, you're going to see things like this, where what's the language of the data? What's the paper reference that you would use for this data? Like the paper that we just shared with you we're using for this exercise. If we go down to, let me see, I'm gonna go back up to occurrence, you'll see what we just talked about. Let's look at recorded by. So a list of names of people, groups, or organizations responsible for the recording the original occurrence. So if you can figure out in the data set who collected this thing, so somewhere in one of these columns is probably a person's name, and that would map, then I have to hit escape, or, no, wait, what have I done to myself? You have to go back and find that it goes in recorded by. So that means when you go to the exercise itself, I can do that now, you are going to find link to narrative exercise, and you're going to go all the way down. This is the introduction to Darwin Core that we just went through. Here's where we're at, looking at this data set, going to the link Katja sent, opening up the quick reference terms. Then you'll open up the unmapped B data set, and you'll be able to then map. Does project map to Darwin Core? Yes or no? Find a term. See if you can find the term that goes with determined by, or the one that goes with maybe Latin binomial. And that is this link to the RCN B data mapping template. So when you open that, you see this. And again, you can do this on paper, on your desktop. Look this up, like look at the data, see if you can figure out what the Darwin Core term is. If you notice a problem with the semantics or syntax don't quite match, you would have to do work to get the data provided into the field, and you can make yourself a note there. And any questions you have about what you see inside the data. So I'm not sure what our time is. How fast did I go through that?
You're doing great on time, Deb.
Yep, we're at 10:48.
All right. Well, I'm just trying to make sure you have, let's see, if it's 10:48, then do we have 30 minutes to give?
Yes.
Okay, cool. We have a few more slides after that, Brianne, just so you know, when we want to come back and discuss. So
great.
Given that, and lots of experience here, we might be able to do less than 30. I'll be pinging all of you. We'll be asking you how far you've gotten, how many terms you have mapped. So this is like ready, set, go. And we're here to answer questions. And I'll be checking to see when you've maybe got about, let's say, 5 to 10 terms mapped. Put a note in the chat with how many terms you've mapped. That would be great to know. So somebody keep time. Thanks. Any questions too, they're most welcome. Let's see if you
If anybody has questions specifically while you're going through it, feel free to raise your hand or throw questions in the chat. As far as where you are in the process, if you can find a term that you think is appropriate or not appropriate, or if there's two that you can't decide between the two of them. As Deb mentioned, the idea of this exercise is not to give a complete overview of every single term in Darwin Core, but really just to introduce where the terms are and what the mapping might look like.
and bear with me too, y'all. There's like 104 comments that happened in the chats while I was giving my talk. So you might have to re-ask your question or ping me again. Link, I just see yours. And I see jump to first unread message, a new feature in Zoom helping me to figure out where in the heck I departed the conversation in the chat and went to slides. So Link, yes. So anybody starting to learn about these topics, this is an intro talk, and that's certainly what we were aiming for here. There are other places online we can find lectures and tutorials, and I'm going to get the audience to help here. Certainly, Karen Goodall, don't have access to the Google Sheet. You, and by say you don't have access, Karen, can you not copy it? You do file copy, file download. I'm not sure if you mean you don't have the link to it, or you mean that you were thinking you were going to be able to type into it. It's a copy you need to make for yourself, because everybody here is doing the same. Yes, you should be able to download the mapping template. Yes, Jeannie. Tell me if you cannot. We just couldn't make it a public one because then all of you would fill it out, and there's 100 people filling out the same form. It's got to be your own individual or group exercise if you're joining. Link, you are having trouble with downloading the template. So up here, so file should be download, and then you can download here in either .ods or Microsoft Excel. Katja, did you want to weigh in?
No, you did exactly what I was going to do.
Got it. Jeannie, if that doesn't help, keep texting us. Keep sending a note in the chat. Because we can also probably find out what the issue. Okay, good. Glad. Thank you for letting us know it helped. So Link, back to your point, there are talks at IDIC Bio. There are talks at GBIF. I think there are GBIF people in the room. The EML link is restricted. So, for the EML mapping, we're not quite to that one yet, but you're ahead of the game. Yes, it is. They're all restricted, but they should be this one. So the EML file, you should still be able to download a copy the same way. For me, we're not doing the EML yet, but you can, the EML mapping spreadsheet. Okay, refresh that, try it now. It should be okay now. Thanks, Elizabeth, and thanks, Karen. I will go and see if that was the other one. Let's see if that's what's going on here too. Oh, anyone with the link can view. Should be able to view and then download that. Okay, good to know. Good. So Karen, let us know if you now have a link you can use. Yay! Hooray! Success. So GBIF. I know there are people in this room. I know there was a link we captured. Katja, I'll see if I can find it real quick. About other data mapping and issues presentations that you can use. Let me see. I think. So just off. So at link. Oh, wait, I forget that we have threading now in Zoom. This is pretty cool. Go back up to your question, like if I can find it. This one.
GBIF there's a lot of resources depending on the question. GBIF does have a quick start guide to publishing data through GBIF, which includes mapping to Darwin core and some EML. Data One also has a number of great resources for sharing data sets. See if I can find.
Yeah, and while you're doing that too, I want to go back. So someone in the chat noted that they had never heard of such standards until this workshop. And I think at the beginning, as we pointed out, where we learned on the job, that this isn't necessarily, although I think it's getting better, something that's embedded into academia yet across coursework. Because for those of you, you've grokked this, you've understood this importance, it doesn't really matter whether you're in biodiversity or the history department or the humanities or the engineering department, it doesn't really matter. Data standards are relevant to what you do. And so this particular, I'll put the link in the chat, and Katja referred to this in her talk. So I'm going to come back to the chat and put this in while you guys are trying to map, and hopefully somebody's keeping time. Check out this paper. And note in this paper, this was a survey, and again, this is 2012. Yeah. So they went around and they looked, I'm still sharing my screen, right? You guys are seeing this chart? Yes. An error. What? Don't copy, just click. Shouldn't be an error. Let me see.
The parentheses, yeah.
Okay, but click on it, don't copy it. I just clicked on it, it worked, but I can do it without the parentheses again. If you try to copy it, it's gonna fail because you're gonna get the dot. There you go, try that, should work. I hope it's not, yeah, it's open access. All right, so in this paper, you'll find this graph. So what happened was they interviewed ecology and evolution departments, universities, and asked them, do you teach these skills in your ecology and evolution departments? So remember I mentioned that data standards help with quality control, file naming, what to use, what software is out there, what makes sense to use for what you want to do. What about metadata? What about protecting data? What about archiving your data? Where is it? Is it safe? Do you have multiple copies? Do they teach you about data reuse and the potential for your own impact metrics by supporting reuse of your data? Data sharing, aspects having to do with reproducibility, they're based on your own choices. These are things, the black bars show you that roughly less than 30% of the departments were teaching these things. So hopefully that, and yes, Link, the point is all of this stuff is applicable no matter which field you're in. It's not just about our realm. But this is showing you that the needle is moving, that many departments were not doing any of this, and that goes for other departments on campus as well. So. How's the mapping going? Anybody have a question or an issue? Or not yet? How many people have mapped at least 5 terms?
Some people are commenting
Yep,
that it's harder than thought it would be. Yeah, yeah. Some 10 terms. 5 thumbs up.
Cool. And again, and thank you very much. Well, who knows, James, what you can look. 6 feel uncooperative. Yeah. Again, the notion here is that you see where everybody is, and y'all can tell already I'm very interested in sort of community transformation and community culture change. So here, when you give the gift of sharing your progress, you're helping us all understand the landscape, because it is hard, especially the first time you do this. It's not just about finding the term that has a similar name that seems like it might be right, but are the semantics right? Is the syntax right? How am I going to know for sure? It's not always that easy. And so somebody asked the question, yeah, like, why not from the start? Well, clearly it's much easier if you know about these and you do this from the beginning. After the fact is definitely harder. So you can imagine you find somebody's data set, you're very excited to use it. And I'm sure many of you've had that. And then you open it up and look at it. Then you have, Yeah, Tom, that the idea was not necessarily the first 10. It was to do as you saw made sense to you. Lots of questions for the data authors, for sure. And Andrew is done-ish. Go, Andrew. So I think just, what do we have, about 10 more minutes left in our 30 minutes?
Yeah,
right?
I think, Andrew, to Andrew's point, though, what I read in that too is that the data authors and the authors of the data sets themselves end up being the best people to actually do the data mapping.
Yes.
Because as the author of a data set, I know what I mean by a certain field at the time when I'm doing the mapping or when I'm collecting the data, and I know how I've collected the samples and where those samples are deposited and things like that. So it becomes much harder the later the data set gets mapped.
And in this case, Andrew, to be very fair to the authors, as I pointed out, they worked very closely with the collections and with the collection manager here at INHS. And I didn't show you the mapped data. So Tommy McElrath worked with Brenna to map the data to get it into our software here, TaxonWorks, and then out the door to GBIF. So that's exactly what Katja just described. Had to happen, though, to go back and forth. Tommy couldn't do it either, looked at their data and could easily map some things, but not everything. And so that conversation was super important and couldn't have been done without it. Yes, yes, and I will mention some of that as well. And for those of you who are working ahead in the tutorial, you'll see a reference to the good data that would be nice to add records if we can. Yes. And no, Andrew, and I didn't mean to frame it that you suggested such a thing. What I was saying is that it was a collaboration to do exactly what Katja said, was that in order to get these data out the door and mapped, it was a collaborative effort between somebody who knows the standards quite well, who works in collections and who supports the use of the data by getting that data out the door, mapped standards, helping the researcher to do the same. I'm assuming by now everybody at least has 5 and probably getting close to 10. So maybe just a couple more minutes. It would be very interesting, Jen, for if you'll share, like, for example, which ones felt uncooperative. And yes, we'll talk about any bugs seen, and anybody else who had an observation about any issues they saw in the data. Those are we're going to be asking you about that in just a second. Mhm. Mhm. Yep, Paulina, we're going to talk about that. I also found those tricky, and I can explain what the author of the paper did along with the collection manager. Yep, Mohammed is agreeing with you. Those were tricky. Do y'all know how to raise your hand in Zoom using the little Zoom icon thingy? I was wondering if we can get a show of hands of how many people are at least half done, because I think maybe we could wrap it up a little bit early, maybe. See hands going up. Yep. Thumbs up, hands up, either one. I guess thumbs up makes more sense, doesn't it? Yep. And some people put a thumb up in the chat. That's cool. So I'm getting the sense that we're probably at a good place to move on to finding out what people did. Give it another minute and then we'll stop. right, I think we've probably gone far enough now. Enough people have gotten the hang of what this exercise is like. And I want to emphasize that this is something, if you have regular templates and you were to have your own, that you do this exercise once, and then you only have to edit it if you're going to add a new term, or Darwin Core or other standards adds another concept that you can use. But you do see that it takes time. So one of my colleagues tells a story about working with microclimatologists, and she's an entomologist. And it took them a year to figure out their data collection protocol, because you can imagine these people collect data in the microseconds that are microclimatology focused versus entomologists who hang a trap out and come back a week later. Very different timescales. So trying to understand what terms they were going to use, what standards they were going to map to, took a long time. So this planning that Katja was talking about could be key to saving lots of headaches, but also to this concept of fair, citable, attribution tracking. All of these things are sort of benefits of going through this work. So let's see if we can go back now and see both how far people got and what questions they have. And let me see. First of all, where does date go in Darwin Core? Anybody could type it in the chat, or where does date go? Ah, there you go. Quick typing, James. Right. And then what about the format of the date? Is the date in one of the suggested formats, the ISO date formats? If we go back and look, I can pull that up for you so y'all can see it. Sorry, I got to find my mouse. There we go. If we go to here, and I've got it blown up really big. Let's see. Control F, event date. There we go. If I go to event date, it says recommended best practices to use format that conforms to ISO. And we can see here, these have timestamps. Or we could have a date that has an interval, or we could have a year interval. And of course we can have, but we notice that it's year, 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 month, month, day, day, with a dash between. And if I'm not mistaken, our sheet has slashes. So you would have to, yep, there you go. Kristen got it. That's right. Yep. So the semantics, the meaning matches. You've got a date and it's the event that the thing was observed, but the format, right? The syntax isn't quite there. And yes, Jort's pointing out the ranges and how those would be supported. But the point is you'd have to do a little bit of work if you're going to put this data out to aggregate with other people's data. That's one example. Let me see
And
here.
this is actually a really good example where early thinking or planning about the format of dates can help with later aggregation of those dates. So that if you start off with the dates being in the spreadsheet formatted in a way that is easy then to transfer or to format into the standard, then it just makes life easier and less mistakes.
And phonology, of course, again, these people who want to do a lot of phonological research with the kind of data we're talking about here are very happy in that. Tom asked, should we put the other format into verbatim event date? Yes. I would always suggest that if you have a date that is in some other non-standard format and it is there for you as evidence, that you capture that, because that helps in case there's any question in the future. Maybe you interpreted somebody's data set as month, day, year, and it was really day, month, year. Who knows? I don't know. That could happen. And having the verbatim could help. What else? What about Latin binomial? Where does that go? And I know there's lots of people in this room who know the answer already, so maybe you wait and see if someone else who's new to this pipes up. Where does the Latin binomial go? Aha, yes, Muhammad, scientific name. Does any work have to be done to get it in there, or does it match what the rules expect for that field? Is it good to go or not? We can look again. Where is it? Where did we go? In scientific name, I think it says something about the author is recommended. There it is. The full scientific name with authorship and date information if known. So you can just put this, but clearly with authorship is better for all the reasons that people in this room know. And it gives you valid examples of what you would expect to find in that field. In this particular data set, we don't have authorship, I don't think. So that's a tricky wicket. Katja, what would you do in that case? I mean, I guess for some B names, it wouldn't matter. It would be unambiguous, but for others, not so much.
I think it's most appropriate to have the author in the year because it, shows that the understanding of the scientific binomial based on that author and year.
Mm-hmm.
But as you mentioned, the standard, that's not an enforced part of the standard, that a scientific name without the binomial is also possible. I do want to mention, though, that scientific name doesn't have to be species or even genus, but it could also be whatever the lowest determination is for that, right? Even to order.
Yes. Are there other fields people want to ask about? Yeah, Kristen, project's a good one. I'm not sure. I think data set name, maybe. But honestly, I myself would need to know what a few more of those fields mean better. Yeah, project, block, and treatment. So these are the kind of things that, why isn't there a verbatim? Okay, let me back up, sorry. Block, treatment, so I can answer a few of these. Again, metadata, block, treatment, etc. If you went and read the paper, some of that can be teased out by you simply reading the paper. But it would be a lot easier for you, right, if those were compiled in a metadata form, where you wouldn't have to go hunting through the paper to see if you could figure out what they mean. And in the paper, I believe you actually see the blocks. They actually have a map, and they have block, whatever that is, block B, block whatever it. But it's not anywhere in a form for you. You have to read the paper to figure that out.
So to add to that, I
Yes.
think what this actually really points out is some differences or potential differences in the way one might think about the metadata for the data set versus what's actually in the field and shared with Darwin Core. So the metadata describing the data set could be very rich with information about how the treatment, how the survey was done, the specific location that it was done, who was involved in that survey, the actual sampling protocols, links to publications even about those sampling protocols. So very verbose information where the data itself mapped to Darwin Core could be less verbose. Here's a good question, Deb. Yort asks, I'd like to understand what SILA means in flower associate.
Yes, and right now, honestly, I had to sleuth this out as well, Yort, and I bet you there are people in this room who already know the answer because this is something that they probably do. So there is a accepted practice that when you are going to note like a host or whatever that like, so this bee was found on this species, they use the first two letters of the genus name and the first two letters of the specific epithet to make a 4-letter code. And this tends to work most of the time. But of course, it's like inside knowledge. And I think I remember in the paper somewhere, and it might have been in the supplementary file, I just tested my thought. In other words, I looked at all the taxa for the flowers that they said were associated, and I said, okay, this is RANI, and then I went and looked in the data set to see if RANI had the scientific name that would match RA for the first two letters of the genus. And so it wasn't written down anywhere. It was me knowing that that's the way that this community tends to do that. So that would also be good to have in the metadata. Yes, abbreviations used would be helpful. And then someone may have, somebody asked me, so I've showed it here on the screen, right? You guys are seeing my screen. So this is the associated taxa field in Darwin Core. And maybe Katja, you could briefly add on to this because I'm going to lead into the next idea of what might be coming next. So here, if you're out in the field, if a botanist is collecting plants, they might say that there was a bee around and they might know roughly Hymenoptera. They might not know anything else, but they mention it. Or they mentioned other associated flowers they see around. But here, of course, with entomology, you often say what it was found on, found with, what it was eating, what it was laying eggs on or parasitizing. So in this field, you can declare the relationship and then the taxon name. So this is where you would put that field in our data set that's called flower.association.
Yeah, and so that relationship in the bee community could be visits flowers of. So a lot of times people on labels of museum specimens will only have the name of the plant because in the metadata, it records that the bee was recorded as visiting that flower if the bee touched the flower or if the bee landed on the flower or something along those lines. But in the associated taxa field, you can actually record that more specifically by saying that it visits the flowers of colon and then the scientific name of the flower.
So Katja, where do people find the terms they should use to express the relationships?
Well, that's a really good question. So there is a controlled vocabulary called the relations ontology that has some terminology. And just like Darwin Core is a standard for understanding fields in biodiversity, relation ontology is a method or a way for understanding the meaning of some term, like visits or host. It doesn't mean that the relations ontology is completely comprehensive, but it does have visits as part of it. I think if somebody could put a link in the chat to that. But the term itself is often free text within the, before the flower. But in the metadata of describing the data set, a person can define that term. So you can define it by linking to an already defined term, like in something like the relations ontology, or you could create a text definition for that term to be very explicit about exactly what you mean by visits.
Cool. So those of you who are interested more about that, I happen to know that this is one of Katja's major interests is these relationship and understanding how you document that and where the controlled vocabularies are or are not for these things. And those are also things you would want, if you're interested in that, to figure out how to, in advance, that you can capture. And Katja mentioned ontologies. There are these very specific places where you actually get a identifier to express the relationship, not just a string, that you can be very, very unambiguous about what you're asserting is the relationship between those two things.
And I'll see if I can find it, but on global biotic interactions,
Mhm.
which is a data indexer for ecological interactions, there is a list of the commonly used interactions and their links to those ontology terms. So I will see if I can find that and then throw it in the chat.
Cool. So heads up, I'm trying to save enough time for just a few more slides at the end of this adventure, but making sure, remember I have to scroll through over 100 comments. So trying to make sure if we missed your question, please ask it again. Why isn't there a verbatim scientific name? That's a really good question, Tom, and it's one field I would like to have, and we've asked for it many, many times. It's a long conversation. There's many reasons why verbatim would be useful, and I suspect verbatim for everything. But it brings up the conversation about AI, which we could go down a big rabbit hole. I won't do it now, but if you want to have that conversation later, I'd be happy to pick that up with you. If you want to ask a question like that to the Tadwig group, there is a place, and I'll see if I can give you the link right now, where if I were you, I would post that question and see what the standards community has to say to you. So I would go here, if I can remember the acronym. Let's see if I have to remember the URL by heart. Here we go. So if you don't have a GitHub account, you'll have to get one, but it's not hard. I would go there and post your question. So what I would do is go to issues, which is here. Oh, good, Abby. Thank you. Thank you. So I would go to issues and click on new issue. And this is where I would say, why isn't there a verbatim scientific name? And then write, I think there should be one because this would help me do X, Y, or Z, and see what people have to say. There was this one in the latest Darwin Core commentary review. So let's say you find a problem with Darwin Core or other data standard from Tadwig. You find a typo in the definition, or you find a part of the definition that doesn't make sense to you, or you find a gap. I have data, I want to share it, but there's no matching term in Darwin Core or audiovisual core or whatever. You can go and post an issue. So Abby posted the Darwin Core commentary review. Once a year, Darwin Core goes through a review, and people can add new terms or ask for revisions to old terms. They can deprecate terms, they're outdated. And you can click there and follow through Abby's point. A lot of Russian doll nesting of PI's institution collection project dataset deposition. Would love some guidance on that front as well. I think asking the EML person and thinking about what I can tell you, James, in the future, GR Cycall at GBIF, this having collections have unique IDs. There are these things called RORs, and I forget the acronym honestly, but it has to do with organizations having unique identifiers. So once more of us have those and they're in play, then it'll be a little bit less confusing, this Russian doll nesting problem, because at least we'll know which part of the Venn diagram we're talking about a little bit better, rather than having just a string, right, for a given institution or a given collection or a given project. That will help. And I'm happy about any of these topics, like Deb said, ROR, what the heck's that? Remind me and write it down somewhere and we'll send you a link, talk more about it. Are there any major questions? Thank you, Abby. Yes. Anybody here interested in, I hesitate, but anyway, briefly, in AI for digitization of data? And if you are, Abby, could you do me a favor and share the verbatim label ticket? I encourage everybody who's interested in the verbatim conversation to read that ticket and scroll all the way to the bottom. There might be some inspiration for you there. Individuals need to be able to receive institution codes and free access to publish their data via the IPT. Individuals versus organizations, you mean, like? That's what you mean. Yes, that's an ongoing topic. Does anybody here from GBIF want to comment on that? Yes, thank you. So Abby just posted one of my topics at the moment that I like is this notion of verbatim label and why that's powerful and how AI can help us mobilize these data. If you're interested in that topic, click on that link. Did I miss any major questions before we go back?
No, I do think just an added point though is that our templates and the way that we share data is evolves. So I think what's important is to in the first pass put the data out there available and as linked to Darwin Core as possible or mapped as possible. And by putting it out there, feedback comes back about how understandable the data is. So there isn't a perfectly mapped data set, I think, on the planet. And again, the standards themselves are evolving and the utility of the data sets are evolving. So that's an important thing to remember because it can be a pretty deep well in understanding the best ways or best practices for doing something. But the first best practice is trying to do it.
So with that, I think it's probably time. And if we missed your question, you do need to let me know. Yeah, and Brianne's posting me in the back. Thank you, Brianne, for that backdoor note about the time. We have, I'm going to make a few more points, and then we have just like 4 more slides. And we want to open it up to more conversation. So let me point out a couple of things. If you open this link to the exercise tutorial, and I assume all of you have that now, and you scroll down, you'll notice that what I did was gave you what it exactly looks like, and then I put answers from instructors. So here you can go through and see a lot of people mentioned project. Was there a Darwin Core term that matched exactly? No. I'm not sure where this could go. I mentioned occurrence remarks, maybe the EML file, and somebody suggested maybe data set name, perhaps. I think it depends, and it's going to depend on your own project and what you're going to do with that data set name. There are other Tadwig folks here. Feel free to weigh in. Would you put project in data set name? Going down, for example, I brought up date for all of you and you said, yes, there's a matching term called event date, but the format's not what we need. It needs to be year dash, month, day, day. So we can all be happy with our past selves. Site name, potentially it could go in verbatim locality or Darwin Core locality. I need to know more about the data set so I can figure out what to put in the locality field. The coordinates fit. You could put the coordinates into the Darwin Core term verbatim coordinates. Did anybody notice this? +1 if you pick that up. False precision. Is that a funky term? It might be new to people. False precision. If you go and look at this data set, look at the coordinates. I don't know how many decimal places that is, but it's a lot. And it's hard to explain to people, but you have to trust me in that data goes from your notebook or your GPS device into a spreadsheet, then into a database, then out of a database and into a spreadsheet, and then into mapping to another format, and then through some other script to get uploaded into GBIF or IDIC-Bio. And let's just say that things happen to coordinates when you do that. And sometimes you get this false precision. And so there needs to be a way to get a reality check there at how many decimal places are really reasonable to think about here. Katja, can you add to that? What should people think about when they see this false precision? Chopping off where? How would they know?
Yeah, I mean, I think we're going to have a talk, I believe it's on the second day, about geospatial standards. And so, but it's definitely something to think about at the time. I mean, ideally at the time that the data is being collected, that there is such a thing as false precision. And there's also fields in Darwin Core that are supported and encouraged, including precision, about the precision and also the uncertainty of that precision in the data.
Okay. All right, so back to this. What else did I want to make sure to mention? I think you mentioned collection ID, and this is a tricky one. So for those of you, and I'm not sure how entomologists do this or what you call it in if we're botanists, our collector number. So if I go out and collect a plant and it's my 200th plant, then I'm the collector and I have a number 200. And in this case, I was able to deduce, I think, that in this data set, collection ID maps to that concept. But again, how to know this without personal knowledge of the data set? That was a bit tricky. I don't know that I would put commas in my barcodes, but for some reason here at INHS, some of the collections do. That was only my comment there, because that could cause problems in the future with matching with or without comma. What else? Again, sampling protocol, probably trap would go in there. But again, how did I know that? I don't know what position means. This was one I noticed that in the data set, they offered AM or PM. And that could be important to all of you who are trying to figure out when to go monitor the bees that are out that you're interested in. There's no field that matches perfectly, but one of the standards folks suggested to me that event time could possibly be used if you were willing to put this in the bucket for AM. So it would be clear that AM is sometime between midnight and noon, and PM is sometime between noon and midnight, although that may not be useful from a research point of view. I have no idea. But I don't know if the data set itself, if you read the paper, AM and PM could possibly be described somewhere. I think I'm pretty much done here. I wanted to point out a couple more things. We didn't have time for this, but something for you to think about. These are fields that could potentially be added. So because you have verbatim coordinates, you can tease out the decimal latitude and longitude. You can tease out the institution ID because it says in the paper that the specimens, and it says in the data set, INHS, and that's what that maps to in Darwin Core, institution code rather. Catalog number, this again, because Tommy worked with Brenna and he gave her numbers to use for those specimens in advance of doing the work. And again, I'm going to allow you can go through and read and ask us more questions. Somebody mentioned taxon rank. That could be added because you can tell what it is of the name used in the identification. You can deduce preparations, individual count, and life stage. And someone, Link, I know you and several others are talking about the citation attribution credit. There is something you can do right now in Darwin Core that at least helps at the level of the individual record. So since all of these records are associated with this publication, you can share this in the record set so that this citation is in every record for one of these bees. Michael asked about a website for registering and looking up institution codes. Yes. Someone please, Abby, are you still here? Or Jen, or
I'm looking for it.
Yeah, Abby will probably pop it in there, lickety split. But there is a place where you can actually find institution codes or get your institution in there if it's not in there. Here's the issues. Oh, I was talking about troubles with finding these, as some of you also had, like, where would I put this information? I can tell you locally, a lot of this stuff was aggregated together, was concatenated, and then put into a locality field inside our database, and some of it inside TaxonWorks. But it was, you can also, I'm happy to share our personal mapping. Now I want to talk about metadata for just a minute. Many of you went on to do the metadata. I'm trying to keep up with the chat, y'all. You can see that's me looking back and forth. Are there any questions I missed, Katja, that you saw? Thank
No,
you,
I think
Abby.
that's-
Sorry.
Yeah.
No, it's good. Yep. Okay, cool. So let me go briefly then back to the slides. And I'm going to switch screens again, right? There we go. So thank you very much. Now, please in the chat, and you'll have to tell us what it is you're sharing in there. One thing you learned, one thing you might change in your future standards of practice, or something you might teach differently if you're in the business of teaching others to do this kind of work. So please in the chat, share one thing you learned, or one thing you might change, or something you might teach differently. Your turn. You learned about plant codes. Cool. You're welcome, Kristen. I learned something the other day, Kristen. We talk about Darwin Core a lot when we're teaching this, but I was in a room full of about 60 people and I asked them if they knew where Darwin Core came from, and they said Darwin Core. I said, well, it comes from the Biodiversity Information Standards Organization, TADWIG. So yeah, good to know that we're able to make these connections, and now you know what Darwin Core is. Yay, Jeannie. Aha. Cool, Eleanor. Yes, Darwin Core versus EML. Yay. And there are other data standards. And if you find gaps in these, let the EML people know, let Darwin Core know, talk with us.
Karen had a great idea about getting working with graduate students to make a DMP, a data management plan, as part of their proposal too. That's a great idea.
Thinking about that, graduate students often have a lot of specimens when they finish their research, and those need good data. So helping them understand the importance of the value of those specimens being enhanced by the data, and helping ensure that they have plenty of time to gather that information and get it there, and help support them if they don't quite know what to do, so that when they finish their projects and walk out the door, those specimens don't end up in the kids' drawer in the collection. That those specimens also, like the data, become valuable for the future. But it's hard to know all of this stuff and what we're out, but we are here to help you. Cool, there's lots more coming in. Yes, metadata. Yes, always. And that's like, keep the raw data raw and make your changes, and always track everything that you're changing. Always keep that record. Good question, Karen, about when to publish data sets, before or after. And the answer is probably it's going to depend. What does it mean one can put Y? Put a Y to voucher. Yes, I have no entomology background. If this is a bee now in my lab and it has a unique ID that I can find where it is, does it mean the bee is vouchered? Yes, I went. In this sense, vouchered does mean in a collection where I could go and find it if I had the identifier for it. So you put it in a collection and I know I can go there and locate it. Katja, do you have a different answer, or?
Well, so tomorrow, Erica Tucker's leading a conversation about vouchers and museum specimen collections. And so that would be a great conversation to listen in on and hear and learn more about. But
Okay.
I agree, Deborah, that the idea of voucher in this case is that it's not that you can find it necessarily, but other people can also find those specimens in order to be able to look at your identifications.
Yeah, you raise an ancillary point. Is that a preserved specimen? So in this sense, yes. However, there are lots of people who, and in some cases we do this anyway, where you could argue that an image is a voucher. So if you could imagine that a species could be identified from an image, if you can imagine that a herbarium sheet or an institution, God forbid, flood, burn, or otherwise specimens are destroyed, the image of those sheets is certainly a voucher that that entity existed on the planet at that time in that place. It may or may not be useful for all research purposes, but it certainly is a declaration or an assertion that's valuable to say this thing occurred and was noted. I hope that answers your question there. Are there fields that can be added to Darwin Core? Excellent question, Jeannie. There are extensions, which we didn't talk about at all. I mentioned very briefly in my talk about things like genomic data. There are extensions to Darwin Core, and if you discover you have data to share that isn't in current Darwin Core, there is another standard called ABCD, and there is certainly the opportunity to add terms to Darwin Core through the Biodiversity Information Standards Organization. So, all right, a couple more slides and we'll be done. So some of you took on the metadata exercise. And I think in the interest of time, we're not going to step away for 5 minutes, I think, and take this on directly. What I am going to ask you, you can follow the link. If someone can put the link to this sheet in the chat. The question I would have for you is, what fields could you populate if you looked at it right now? And if it were you, what would you want there in that form? So imagine you're trying to evaluate someone's data set and decide if you want to use it or not, if it's valuable to you or not, if it's worth your time or not. Take a look and see. Yes. Oh, Abby, thank you for sharing the extensions for the person who asked about, are there fields that can be added to Darwin Core? Yes, indeed, there are. And what I'm talking about is, Katja, could you share the link to the EML mapping spreadsheet?
Yes.
I think I just put it in the chat. Is that the right
Oh, you
one,
did.
Deb?
Oh, yes. Excellent. Thank you. I just didn't want to click away from my slides and then I'd be lost again. Yeah, so the idea would be here that you get a sense of what Katja was talking about in planning. So is this the kind of stuff that you capture formally when you start your new research project? Is it in your head? Is it written down in your field notebook? Where are you keeping track of who's participating on the project? Who's going to be the resource so-called creator? Who's going to provide the metadata? How are you going to describe the coverage? Do the members of your team have ORCIDs so that they can be uniquely identified? The project data, right? Who's keeping that and where is it? Often you find it's in a spreadsheet somewhere, or it's in your head or in some document that only 1 or 2 people have access to. So it helps to kind of look at this and maybe democratize the procedure a little bit. And I'm just curious, anybody else want to add to that? How you construct metadata? Katja, how do you know what to collect, what to write down for your metadata?
We usually start in a spreadsheet format, just like you have here, to collect the metadata. And it's a living document, try to answer the questions at the beginning of the study, but as especially the sampling protocol may change over the course of some study, then or additional details for how sampling is done, then the metadata document gets updated. And so it's almost like a how-to document, and even can be incorporated or as part of the research plan to begin with, like how sampling is even getting done or getting started. As Jen points out, the resource contact is a little bit, it's good to have a singular point of contact. So in that case, for resource contact, we usually add whoever is the lead on whatever project it is, which could be the lab PI or graduate student or somebody else.
Cool. So and as Jen points out, not only are people, we are all ephemeral. So that's another reason why these ORCID IDs are nice, because it helps you understand in a longer context than our lifespans about who did what with what data and generated what knowledge and information. So thank you, Katja. I would say, let's see now.
Deb, you have
So,
about 5 more minutes.
right. So I think, and I only have like a couple slides here. I think all of you now see that in general, this notion of making information fair, this findable, accessible, interoperable, and reusable. I mean, you can put your data somewhere where someone can find it, but how accessible it is and how reusable it is have a lot to do with have you taken care for this step to get it mapped to data standards, make sure the metadata is accurate and filled out enough that someone can use that data with care, that it's used in a way. And also, yes, oh, hang on, Yort. The notion here is that when you do this too, and you share these identifiers for people and you share the data set in a structured way, these are the kind of things where we can begin to try and automate some of the making data accessible and making sure that it's easy to cite. So for those of you who've never heard of Bionomia, this will be a first experience, but we can put in, you can have a look. See, did I get the link right, David? I see. There's a place where you can go and look at specimen data from a people orientation, who collected what, who identified what. And one of the cool things you can do there, if you're a researcher and you have specimens vouchered, is publish that data set that has your intellectual capital wrapped up in it directly to Zenodo, findable, accessible. And so this notion, what's your role in this? Again, data standards is a big part of that, let alone the metadata and other things we talked about. So what kind of help do you need to make the data more fair? And Yort wants me to explain the difference between open access and fair. Katja, I think I'm going to pass that ball to you.
So open access, the way I see it is that that's a term that comes from the publication community, that the data sets or publications are openly available through the internet. And that fair is more specific, that the data sets and information are, because you can be open access and not necessarily findable. You can be open access and not necessarily easily accessible, because you can find because it's accessibility has something to do with findability. You can be open access, but your data may not be interpretable. It may not be interoperable. So, so I think that there are differences but mostly I think of open access as a terminology that comes from the journal publication community specifically, where fair is more about the exchange of data.
So I would be interested when you share, all of you, if we have Link has great questions about data, and we can keep going and continue this conversation, Link, but what help do you as a community need to make the data more fair and make sure there's something in it for you as a result, which I would argue is the sort of reuse citation attribution conversation, but what help do you need? Would be good to know. This comes back to you. You have the power to take this data that you collect for your purposes and expand its reach and its lifespan. This also will affect you and your impact metrics and that of your institution that helps support you mobilizing that data and the funders who provide the money to do that, which is often the case that they're asking for these kinds of things. So facilitating data use and reuse, facilitating the life of the data, all of these things should help you with your research and your research productivity. At the same time, should give us ways to help facilitate the credit citation attribution conversation in a positive way, I hope. Katja, final thoughts too?
Nope, that was great, Deb. Appreciate your presentation.